Hello everyone, and welcome to Chapter 8 of Microbiology. In this chapter we're going to talk about microbial genetics. So a lot of this is going to be review, some of the early stuff as far as information about what genes are, the nucleotides, nitrogenous bases, double-stranded helix, and then some of it will be just very specific to microbes. So a little bit of it you will probably feel comfortable with, and then some of the other stuff that we do will be a little bit different. So a little bit of terminology, I won't spend a whole lot of time on this, but genetics is just simply defined as the study of genes and how they carry information and how that information is expressed. A really important thing to remember about the study of genetics or genes in general is that the genes carry the information on what the characteristic or trait should be, and it's the job of the proteins to carry out those wishes. So the proteins are going to express the wishes of the DNA. So that's also very important to remember. The way we define the gene is that it's a segment of DNA that encodes a specific functional product, which is primarily a protein. So your genes are part of your genotype, and your proteins express your phenotype or your physical characteristics. Chromosome is a structure containing DNA that physically carries that hereditary information, and these chromosomes contain the genes, that segment of DNA that codes for a specific functional product, which is usually a protein. And then when we talk about the genome, we're talking about all of the genetic information within the cell. So what we're looking at here is that we're looking at the chromosome that has been disrupted in an E. coli cell. So all of this stuff that you see just kind of squiggled out everywhere, that's the chromosome itself. So that's all of this cell's genetic information that has been disruptive and is no longer in that nice double-stranded helix that we like for it to see and all compound and compacted together within the cell. So just to give you an idea of sort of the dimensions we're talking here. So before we start to talk about any of this sort of microbial genetic stuff, let's kind of review the flow of information. So what we have is we have the parent cell, and we've learned from chapter 6, I believe that was, that bacterial cells replicate via binary fission, where basically one cell splits and becomes two. So the flow of genetic information, and we'll talk about these different ways on how that information can be transferred, is that we can have horizontal gene transfer or what we call vertical gene transfer. Vertical gene transfer is going to be the exchange of information between bacteria that are in the same generation. Horizontal gene transfer is going to be the flow of genetic information from parent cells to daughter cells. So with expression, this is just the DNA expressing the physical characteristics of the DNA via the proteins. In order to do that, we used an integrated process known as transcription and translation, and we'll talk at length about that in this chapter. In recombination, this is where genetic information can be transferred between cells in the same generation. So this would be, recombination is an example of that horizontal gene transfer that we just talked about, that cells within the same generation are swapping information. Horizontal gene transfer, vertical gene transfer would be going from parent cells to daughter cells. And then replication is an example of this vertical genetic information or vertical gene transfer, where genetic information is transferred between generations of cells from parent to daughter. So before we start to talk about any of those other aspects of gene transfer, let's kind of review what DNA actually is. DNA is considered a polymer of nucleotides. Polymer means that it's made of many different working parts. So the many different working parts that make up this polymer of DNA consider nucleotides, which can be A, adenine, T, thymine, C, cytosine, or G, guanine, and then you also have, and those are our nitrogenous bases, the A's and T's and C's and G's. And you also have a 5-carbon sugar called a pentose that makes up part of the backbone of DNA. And the other monomer that makes up DNA that helps to make up this polymer is known as phosphate. So there's a sugar-phosphate backbone that we can associate with DNA. It's a double-stranded helix that's associated with proteins. As I said before, the backbone is the sugar phosphate backbone, and at 5-carbon sugar, the specific name in DNA is called deoxyribose. In RNA, that name of that sugar is ribose. And the strands what holds together the A's partnering up with the T's, the C's always partnering up with the G's, very important. In fact, I'm going to put a star here to know that in DNA, A pairs with T, C pairs with G. What holds them together are hydrogen bonds. So there are different numbers of hydrogen bonds between adenine and thymine than there are between cytosine and guanine. And the way that these strands orient themselves is that they run what we call anti-parallel. So if one strand of the DNA, here's my sugar phosphate backbone, and then the rungs of my ladder would be A pairing with who again? Pairing with T, and C pairing with G. And we know that I'm not going to draw them, but there are double bonds that hold them together. The sides of the backbone are that sugar phosphate. So deoxyribose phosphate, deoxyribose phosphate. And if one side of this strand starts off with this 3' where the phosphate group is connected to the 3rd carbon of that 5-carbon sugar deoxyribose to 5', then the other strand will run 5' to 3'. So that's what's meant by anti-parallel to one another. It's almost as if the strands are running upside down. One is right side up and the other one is upside down. The way that we copy DNA is that we're going to follow it in a 5' to 3' direction. And when we copy that DNA in a 5' to 3' direction, we do it with a special enzyme called DNA polymerase. So important that I'm going to highlight it. So DNA polymerase, you can tell it's an enzyme because it ends in ACE there. The job of DNA polymerase is to add on additional nucleotides as we're trying to separate the DNA and make an additional copy of it. So DNA polymerase's job is to put new nucleotides on here, and it's the job of the primer to initiate this whole process to make it happen. Since we have the DNA is double-stranded, we're going to have a leading strand and a lagging strand. The leading strand is always going to be synthesized continuously because it runs in a 5' to 3' direction. Remember on the previous slide where we talked about the strands running anti-parallel from one another? Well, the strand that's kind of upside down in the 3' to 5', it's not going to have a continuous mode of replication because we can only add new nucleotides on there in a 5' to 3' direction. So we call that our lagging strand. So in order to make our lagging strand, it kind of synthesizes itself or makes this new strand discontinuously. So what we're really talking about now is how we have DNA in one bacteria. We want to separate that DNA, make a copy of it so that we can have new daughter cells in the next generation. That's really the big picture of what's happening. We're just looking at it on a molecular level as to how do we get the DNA to make a copy of itself. So that's what we're talking about. So those little fragments that we're left with in the lagging strand, because it synthesizes itself discontinuously, we call Okazaki fragments. The RNA primers are removed and the Okazaki fragments then joined together by another enzyme along with DNA polymerase. Kind of goes in 5' to 3' direction. That's the only way polymerase can go. But then we have another enzyme, DNA ligase, which will sew all those strands together. And there are several animations that will showcase this, and we'll look at some of those in class as well. And we'll, if we have a chance, look at some of those in this session, some of those animations that illustrate this. So we've already come into contact and we have discussed 2 enzymes already that are responsible for DNA replication. But these are all enzymes that are responsible for expression and repair. Those enzymes that I highlight are important enough for you to commit to memory. So DNA polymerase, which we talked about, you should know that, synthesizes, proofreads, and repairs the DNA. DNA ligase joins DNA strands, joins those Okazaki fragments, so those are the 2 that we talked about. We have helicase, which unwinds DNA. We also have RNA polymerase, that's very important. It copies DNA copies RNA from a DNA template. We haven't talked about it yet, but we will soon talk about that. And then there are what we consider endo and exonucleases that will cut the backbone of the DNA strand and facilitate repair and insertion. And then exonucleases can cut DNA and also facilitate repair. So those that I've highlighted, those are pretty important that I want you all to remember. So here's a pretty picture of what we talked about as far as that replication. So this would be an example of that vertical gene transfer. Notice that we are going from the parent generation to the daughter cells, or to the next generation. So one cell becomes two through that binary fission. That you all are experts at. You completely understand that. What we're talking about now is what happens on a molecular level. So with that DNA, we have our enzymes that unwind the parental DNA. We have our end that's 5' to 3', and our end that's 3' to 5'. Remember, DNA polymerase is only going to go in one direction, 5' to 3'. So this 5' to 3' strand has to be our leading strand. And notice that when it's time for replication to take place, that it goes on continuously. There are no breaks on this leading strand in the DNA sequence. On the lagging strand, on the other hand, we still have DNA polymerase moving in a 5' to 3' direction, but it has to do so in short segments. And these little short pieces that we're left with are called Okazaki fragments, and it's the job of this enzyme DNA ligase to join those fragments together. So here is just another version, another picture of how we look at this circular chromosome. And we have this point of origin of replication where they start to separate the paternal strands from the paternal strand separating to make the daughter strands, and you have these points of replication where it separates out, and the DNA, the double strand, gets pulled into two single strands where you have a leading strand and a lagging strand, and we add the new nucleotides on there, and we call those points of separation the replication fork. And then at the termination of the replication, we're left with two daughter cells that are genetically identical. So now we're going to talk about some more gene expression. We very briefly talked about replication in a type of vertical gene transfer. Now we're going to talk about gene expression. So if we go back to our original roadmap, we've done this part. We talked about replication. Now we're going to talk about expression. And expression happens in two steps, transcription and translation. And as we said before, this whole aspect of expressing the genes is usually the job of a protein. So you can kind of think about the DNA as being a crime mob boss. And if you're a crime mob boss, do you actually have to put the hit out on someone? Of course not. You pay your thugs, your goons, whatever you want to call them, your, henchmen, to do the job for you. So it's the DNA is the crime boss, and it puts out the wishes of what it wants to have done, and it's the promoters that are the workhorses. So in transcription and translation, we are giving the instructions on how or what specifically these proteins should do. So the first part of gene expression, the first part of, another way to say this, protein making, is that we have transcription. In transcription, the DNA is going to be transcribed or written in a slightly different code. It's going to be written to make various different types of RNA. The first type of RNA we'll make is messenger RNA, which has the message of the DNA, and then we have transfer RNA. That's job is to transfer the correct amino acid sequence to match the codon on the messenger RNA, and then we have ribosomal RNA, which holds the whole complex together. All of these pieces we'll see in just a moment. Transcription is going to begin when RNA polymerase binds to the promoter sequence. Now, if we just look at the term transcription, it says a lot in of itself. I've had many jobs. I've always been one of those people that if I liked a job, I worked it. If I didn't like a job, I had no problem letting it go because I knew something else was coming in the future. And one of the jobs I had was a transcriptionist. I would listen to doctors' orders, and I would type those orders out. Now, as a transcriptionist, am I going to change those doctor's orders? No. The only thing that I did was convert the audio communication into written communication and typing them out. Same exact thing is happening in transcription. We're going to go from the language of DNA to the language of RNA, which is a little bit different from what we see in DNA. So like DNA replication, transcription begins in the 5' to 3' direction. The process stops when we reach the terminator sequence of the gene. So purple strand is our DNA template, and then this aquamarine strand is going to be our messenger RNA. So in transcription, we're making messenger RNA. Big thing to remember. So where there is a T on the DNA, A will match up on the RNA. Where there is an A, however, and I'm going to highlight this because it's important, there's going to now be a uracil. On RNA, there is no thymine. So A's partner, when you go from DNA to RNA, is always U. Now, for thymine, there is an adenine. So its partner on RNA is still going to be A. And if we look at cytosine, its partner is still guanine. And guanine's partner is still cytosine. But notice that A will now match with uracil. That is a big way in which the information, the code from the DNA is changed as it goes from DNA to RNA. So RNA has no thymine. So there's no thymine, no T's in RNA. And remember, that's what we're doing now. We're going from DNA to RNA. So no thymine in RNA, nor does this exist. This does not happen. Thymine does not go to uracil. Only adenine goes to uracil. So we have our promoter gene says, hey, start making this messenger RNA strand here. Then we have a process of elongation where it gets longer, and then at the terminator sequence it says, hey, this is where the genes end to make this specific protein. Because really that's what we're doing. We're making proteins. Now in eukaryotic cells, there's a little bit more modification that happens, and we call this post-transcriptional modification that takes place in eukaryotic cells. In eukaryotic cells, this process of transcription takes place in the nucleus. Not the case in prokaryotic cells because they don't have a nucleus. But the post-transcriptional modification that takes place in eukaryotic cells is that you have these parts of the DNA called exons and introns. The introns are what we call non-coding portions of the DNA. We used to believe there was nothing on there at all, but we've since learned that there is something and some value there, but they're not part of what makes the protein. So the introns are going to be spliced out, and they'll stay in the nucleus, whereas the exons are all going to be joined together, and that's what's going to be our messenger RNA sequence that exits out of the nucleus. So exons get to exit out of the nucleus when they are transcribed onto the messenger RNA strand. The next portion, or the second step of this protein making, is translation. So in translation, if you speak more than one language, you're familiar with the fact that some words in one language don't necessarily translate into the other language. But for the most part, you want to keep the message the same. So we're now going to go from the language of nucleotides, A's, T's, C's, G's, and U's, the adenine, thymine, guanine, cytosine, and uracil, to now going on to talk the language of amino acids to make the protein. So the messenger RNA is the template upon which these proteins are going to be made. And that messenger RNA is going to be translated into codons. So codons consist of 3 nucleotides. The translation of messenger RNA begins at the start codon, and then it's going to end at what we call nonsense codons. And another way to call a nonsense codon is just a stop codon. So stop codon just says, hey, this is the point at which this messenger RNA has received from the DNA the genes to make this protein. And we have 3 nonsense codons. Notice that they have 3 letters. All codons have 3 letters. All codons are also found on messenger RNA. Messenger RNA equals codons. but we have 3 that say stop, and we only have 1 that says start, and that's AUG. So in total, because we have 3 letters, A, T, C, G, or actually A, U, G, C, 3 letters of those 4 make up a codon, and there are 4 letters that we could possibly have to be in that 3-letter combination. As a result, there are 64 codons that are on the messenger RNA strand. Those 64 codons are going to encode 20 amino acids. So another way to think about this process and understanding that these 20 amino acids are the exact same 20 amino acids that make up your body, even though you are vastly different from E. coli, vastly different from a bacteria cell. We say this is a universal language because although there are only 20 amino acids, the number of combinations that these 20 amino acids can exist in is virtually endless. Think about it like the English alphabet. We have 26 letters in the English alphabet, correct? Yes. How many words can you make from just 26 measly letters? Infinite, isn't it? So in much the same way, the combinations we can have from just these 20 amino acids is infinite, and that's why we have such an abundance of different types of proteins and different types of biological organisms. So the transfer RNA is going to carry the complementary anticodon to the codon that's on the messenger RNA. On the messenger RNA, we have codons. So for example, UUU is a codon. On the transfer RNA, at one end of that transfer RNA, it's called transfer because it's transferring things, we will have a anticodon. So the anti of UUU would be its match, AAA, because A pairs with U in RNA. Is going to be the anticodon at one end and then an amino acid at the other. So for this example, the genetic code is made from the codon. So from this codon of UUU, the transfer RNA is going to carry AAA as the anticodon, and then the amino acid that it carries at the other end is phenylalanine. So what you're looking at before you is the genetic code. So this genetic code is all 64 of those combinations of codons, 3 of which are stop codons. Here they are, stop. And then we have 1 start codon. And then we have, for those 20 amino acids, like leucine, proline, histine, arginine, serine, glycine, alanine, valine, phenylalanine. So all those 20 different codons, they are going to be coded for on this list. So I'm not going to expect you to remember that UUU is phenylalanine or ACU is thyrine. I'm not going to actually remember that because I will give you this chart. But I will ask you to go from DNA to messenger RNA to transfer RNA to protein. So transcription and translation, I will ask you to do that. And we'll do a couple of examples of these in class. And I will give you this chart. Now, in bacteria cell, unlike what we saw in eukaryotic cells, the process of transcription and translation happens simultaneously. So it takes place at the exact same time. So when we make our messenger RNA strand, as we make our messenger RNA strand, we're going to translate that messenger RNA strand into the protein, so into the appropriate proteins. So a big thing to remember is that in bacterial cells, this process happens simultaneously. So for the process of translation, we've already talked about 2 of our 3 RNAs. We have our messenger RNA that was made at the end of transcription, and then we have our transfer RNA, which carries the anticodon at one end and the appropriate amino acid for the codons at the other end of it. And then the rRNA, the last one, is a ribosomal RNA that kind of holds everything together. So the messenger RNA is going to be fed between the large and the smaller subunit of the ribosomes. And as it's fed in there, it has a couple of spots to it. It has a docking station for the transfer RNA and an exit station for the transfer RNA. So we kind of just feed this whole process along. So at our start codon, the anticodon of AUG is UAC, and then the codon of AUG is methionine, or the start codon that's there. As we move this process along, then we put the next anticodon and codon combination carried by transfer RNA, and then this codon here, this transfer RNA, will then leave. And we do that continuously until we reach the stop codon. And at which point we have a growing chain of amino acids. Now, we started with our start codon, and now we're getting down here to the stop codon. When we reach our stop codon, this whole process disassembles, transfer RNA goes its way, the messenger RNA is then going to be degenerated. We no longer need that messenger RNA, and the ribosomal subunits are all going to come apart, and then we have our new protein. So at the end of translation, we should have a protein as a result of that. And remember my discussion or our example about the crime boss. Now we have a protein that's able to carry out the wishes of the DNA. So here is a link that I'm going to try to show you, but it doesn't work very well here. But I would like for you to watch this, and it's the process of transcription and translation on YouTube. So gene regulation is the next little aspect we're going to talk about. So keep in mind that not all genes are always regulated all the time. Those genes that are regulated at a fixed rate, we call them constitutive genes. Those genes that are only regulated when we need them, specifically if it's in the presence of certain enzymes or only when we have enough of something, they're going to be regulated. So we have different classes of regulation: inducible genes, repressible genes, and catabolic repression. Inducible genes are going to be genes that have to be turned on. So normally the default setting for an inducible gene is that it's off. We have to turn it on because there has to be the presence of something that says, hey, I need the genes for this protein to be expressed so I can break down this thing or do this thing. Repressible genes are normally in the default setting of on until something represses them, like, hey, we have enough of this particular component, let's turn this gene off. And those are the two types of regulation we're going to spend most of our time talking about. So the first one that we'll talk about is an inducible gene. And a great example of an inducible gene is the lac operon. Inducible genes have a couple of different portions to them. You have a control region of your operon, and this is where we have our promoter sequence on the DNA that says, hey, start transcribing here. We have an operator sequence that if it is open, then we continue transcription to make the structural genes that are needed for the actual protein. If there's something blocking it, then it's going to be turned off, and there won't be anything to make it go further. So in that operon, you have control region, which has promoter sequence, the operator, and then you also have the structural genes. The most classic inducible operon is the lac operon, which stands for lactose operon. So in the lac operon, if there is no lactose present and this organism can break down lactose, then it's normally in its default setting. So the operon is normally turned off when we have an inducible operon. So when RNA polymerase attaches to the promoter sequence, well, there's something there. So it can't get past this repressor protein to make these structural genes to break down lactose because there is no lactose present. So we're saving energy by not making this enzyme, or the enzymes, I should say, to break down lactose because there is no lactose that is present. However, if lactose does become present, then what happens here is that lactose, is also known as allolactose, will then bind to our repressor, and it causes it to make a shape change. So that repressor protein is now going to change shape so that it no longer fits into this operator sequence. Remember before that it fit into this nice little, come on, go back. not fitting nicely. Okay, it fit nicely right into there. And then when we change the shape of it, it no longer fits into that. So now, when we have our RNA polymerase that comes along and it wants to make the messenger RNA strand, it's able to do so, and it will make beta-galactase, permease, and trans-acetylase, which are 3 proteins that are needed to break down lactose. A repressible operon is the exact opposite. So repressible operons are always in the default setting of on. So notice that the repressor protein is inactive. It doesn't fit into that operator sequence. So it's always turned on. Very important aspect to remember. And what turns this off is that we have an accumulation of a particular product that we don't need to make anymore. So instead of continuously making it and it's a waste product, the bacterial says, so you know what, I'm going to turn this off. Classic repressible operon is called a trip operon. So in the presence of tryptone, when there's enough of it on there, it will bind to the inactive repressor protein and cause it to change its shape, or tryptophan. It will cause it to change its shape, and it will bind itself to that operator sequence, so that when the RNA polymerase goes along to make the messenger RNA and transcribe and translate the genes that come after it, it will no longer be able to do so. So a trip operon, which stands for tryptophan, this is an operon that's going to always be on unless we have enough or too much of this tryptophan that's been produced. So at this point, and this is just an amino acid, at this point here, the excess tryptophan turns the repressible operon off so that we don't make any excess of it. Lac operon is our classic example of an inducible operon. All right, so in our next session, we will look at some animations for both inducible operons and repressible operons, and we'll finish up this chapter. Have a great morning, noon, or night.